Morning or good afternoon to you all, dependent on your time zones. My name is Aidan Battison and I am a postdoc research associate here at the DSI Nanomedicine platform at Nelson Mandela University in South Africa. And today I'm going to be giving you a brief introduction into nanomedicines as smart drug delivery systems to combat breast cancer. Now, we know that cancer is defined as cells that divide uncontrollably and can metastasize and spread and obviously infiltrate healthy body tissue, leading to the destruction of those cells and that organ. Now, cancer cells differ to healthy cells such that they grow in the absence of a controlled growth signal. They don't comply to programmed cell death. They can metastasize and spread to different areas of the body. They also promote increased vasculature growth to the tumor site. They can evade immune response and as well as being energy hungry, so taking preference to all the nutrients and oxygen supply that the circulatory system gives to that area. Now, the conventional methods for treatment for cancer include chemotherapy, radiation therapy, hormone therapy, to name a few. And while these methods have been proven to be useful, they often come with undesirable side effects such as weight loss, hair loss, nausea, fatigue, bruising, blood clots, to name a few. So generally just a very poor quality of life for those patients who have been diagnosed with this disease. So in terms of statistics, Globacon 2020 estimates that for both sexes, breast cancer to be the number one form of cancer that has been diagnosed, followed by lung, colorectal, prostate, and stomach cancer. And it's very interesting to note that in 60% of cases of people diagnosed with breast cancer will often have a type of metastasis in the lung. So lung and breast cancer are often very interlinked. Now, in terms of South African statistics, we can see that breast cancer is the number one type of cancer that was diagnosed by frequency in 2021, and it was the third most prevalent cancer to cause ultimate death in the patients. So, breast cancer can be divided into certain subtypes based on their molecular features. So some of these subtypes include luminal A, which is estrogen and progesterone receptor positive, luminal B, which has a higher expression of the cell proliferation marker Ki67. Then we have human epidermal growth factor 2 positive or HER2 positive, which is the overexpression of the HER2 receptor with the negative expression or the absence of the estrogen and progesterone receptors. Then we have a basal-like breast cancer, which is HER2, estrogen, and progesterone receptor negative, which is also known as triple negative breast cancer, which is obviously quite difficult to treat. Now, triple negative breast cancer is a particularly aggressive form of breast cancer with limited treatment options due to the absence of these specific molecular receptors on the cell surface. And it accounts for up to 24% of newly diagnosed breast cancers, and a steady increase has been noted in the literature. Now, there also seems to be an ethnic disparity between patients diagnosed with triple negative breast cancer, such that women of African ancestry are more prone to be diagnosed with triple negative breast cancer as opposed to women of European ancestry. And there are some risk factors that come with this type of breast cancer, such as age, diet, race, as well as the expression of the BRCA1 genetic mutation. Now, this is a graph just highlighting that ethnic disparity of women diagnosed, or just generally of people diagnosed with triple negative breast cancer, such that people of African ancestry, or women specifically of African ancestry, are more prone to be diagnosed with triple negative breast cancer as opposed to women of European ancestry. And for an African continent, an African university, this is quite a problem that needs to be addressed, and this is something that we are working heavily towards at the DSI Nanomedicine platform. Now, triple negative breast cancer has obviously limited treatment methods, as it is HER2, progesterone, and estrogen receptor negative, so hormonal and trastuzumab based treatments have proven to be ineffective. And so most of the common therapeutic methods would include surgery, radiation, and chemotherapy. However, these methods have certain drawbacks, such that we often have the rapid drug degeneration, we have drug resistance forming, we have the passive or the non-targeted distribution of the drug in the system. There is poor pharmacokinetics associated with these approaches. There is rapid biological excretion of the drug, and obviously we still have some of those unwanted side effects that these therapeutic methods cause. And this is the basis of the active targeting or the smart drug delivery system which nanomedicines are able to facilitate. And this is what we study and we focus here mostly at the DSI. Now, a bit of an introduction into nanomaterials. We know that these are very, very small particles that have a dimension, at least one dimension within the range of 1 to 100 nanometers. And because they are so small, they have specific characteristics such as a high surface area to volume ratio, they are highly mobile, and they exhibit what is known as quantum effects. Now, nanomaterials can also be divided obviously into organic and inorganic based materials based on their construction and their chemical profile. And apart from all of the different types of applications that nanomaterials can be used for, we want to use them as active targeting drug delivery systems to specifically target a cancer cell and specifically deliver a chemotherapeutic, a photosensitizer, a drug conjugate, or whatever that therapy might be, specifically to the targeted cell, thus leaving the healthy cells unaffected. This is just an illustration of some of the organic and inorganic nanomaterials that have been used in both breast cancer as well as triple-negative breast cancer therapy. So we have formulations such as mycelials, dendrimers, liposomes, polymeric, as well as polymer drug conjugates in the organic category of nanomaterials. And then we have metallic and carbon-based nanomaterials, usually constituting the inorganic side of the nanomaterial spectrum. So why do we want to use nanomaterials, or why are they advantageous as compared to conventional treatment methods? Well, firstly, they increase the solubility of the drug. They are able to encapsulate that drug and deliver it specifically to the targeted site. They improve the pharmacokinetics. They enhance the biodistribution. They have less systemic side effects, and they obviously have a lower toxicity to the system. So this is quite an advantageous therapeutic approach, that the incorporation of that same chemotherapeutic agent just into or within a nanomaterial can drastically enhance its therapeutic efficacy. So that you can see that I'm not pulling the wool over your eyes, nanomaterials have actually been on the market to treat breast cancer for quite a number of years. And if you see in the slide, we can see that Doxil, which is a liposomal formulation of doxorubicin, was FDA approved in 1995 already. So if there was any discrepancy or uncertainty regarding the safety of nanomaterials as drug delivery systems, that can be squashed and put to bed. So when we speak about active and passive targeting, we need to go a little bit more into what is known as the tumor microenvironment. And some of the tumor microenvironment characteristics are tumor infiltrating lymphocytes, the high expression of vascular endothelial growth factors, we have tumor-associated macrophages, and other molecules promoting the growth and the metastasis of cancer cells. Now, some of the general cellular characteristics include hypoxia, a lower pH or more acidic environment, high interstitial fluid pressures, we have impaired lymphatic drainage, leaky cell vasculature, as well as an increase in the extracellular matrix stiffness. Now, these are differences to normal cells, but they actually provide a targeting approach, or something that's different that we are able to target specifically because it is different to healthy cells. So this provides the basis of that active targeting strategy that we want to employ with nanomaterials. So when we speak about active and passive targeting, what does this mean? Passive targeting is the natural accumulation of that material in the cancer or the tumor site based on the enhanced penetration and retention effect by the cell or the leaky vasculature. So it's just the natural accumulation of those nanomaterials at the site, whereas active targeting is a far more specific and direct approach, such that we are able to surface functionalize a nanomaterial with specific receptors or targeting moieties that will seek out and bind to specific receptors on the cell lines of cancer cells, thus producing a more targeted and efficient therapeutic approach. So it's far more advantageous than just the natural accumulation or the passive accumulation of these nanomaterials in the tumor site. So this is just an image that is showing you some of the the receptors that are expressed on breast cancer cell lines, denoted here by the little locks, and some of these targeting receptors that a nanomaterial can be functionalized with in order to seek out these types of receptors specifically, thereby promoting this active and targeted approach. So nanomaterials have often been described as the Trojan horse of medicinal systems, and this is actually shown here in this image quite. It's quite a cute image. I believe that we see that the hypodermic needles are not able to penetrate or get past that wall. This can be seen as the cancer cell, something like drug resistance, that the chemotherapeutic drug is not actually able to be effective. But as soon as we encapsulate that chemotherapeutic drug in a nanomedicine or a nanosystem, slap an active targeting moiety to that system, all of a sudden the cancer cell just wants to open up and let that nanomaterial in, but not knowing of all of the cancer drugs that are inside. So this is essentially a Trojan horse system that nanomaterials can be compared to in their therapeutic approach. So you can see trying to pull the wool over your eyes. If you go onto the internet and just search nanomaterials and breast cancer, you will get so many hits of how nanomaterials have been effectively used both in vivo and in vitro, as well as studies that are showing new formulations that are undergoing clinical trials for the effective treatment of breast cancer as well as triple negative breast cancer. So here are just a few examples which I'd like to outline to you. In example A, we have an extracellular vesicle which has been functionalized with a drug or an antibody for a targeted therapeutic approach. In example B, we have a liposomal formulation, which was surface functionalized with the estrone receptor, which actively goes and seeks out the estrogen receptor on the cell surface of specific types of breast cancer subtypes. And we can see that it has been loaded with epirubicin as well as paclitaxel within that liposomal formulation for that targeted and effective drug delivery system, both in vivo and in vitro studies. And here we have the last example of a hybrid formulation such that we have a gold nanocore, which has been coated with a mycel formulation, or a mycel formulation has encapsulated that gold core. And what is interesting about this system is it has two types of therapeutic approaches, such that it has the active targeting folic acid moiety that has been surface functionalized on that mycel part of that nanoparticle. But within that mycel formulation, we have a pH responsive hydrazone linker that is linking to doxorubicin. So once that folic acid receptor conjugates to the specific receptor on the cell line that it is actively targeting, because of that lower acidic or the more acidic environment of the cancer cell, is able to release the doxorubicin via that pH labile bond, thus actively targeting and releasing specifically the doxorubicin within the cancer cells that express this lower pH environment. So thank you very, very much for your consideration of my presentation today. I hope that it has been enjoyable. And I think one of my concluding remarks would be that women around the world can rest assured that nanomedicines are doing their part in order to make sure that breast cancer and unnecessary deaths from breast cancer is a thing of the past. Thank you very much for your attention, and I look forward to answering any questions that you may have. Thank you very much.